Hello, how are you?
I'm good. How are you?
I'm hanging in there. Thank you very much.
That's good.
Hope you had a wonderful weekend.
Yeah, I did. How about you?
It was good. Weather was nice here. I suppose it was equally nice in Minnesota.
Um, it was nice, but now it's raining.
Oh, okay. No, it's still perfect blue sky right now. So you can't beat it. It's wonderful fall weather.
Huh, that's nice. Okay, so first we're starting with introductions and then I'll get into questions. Do you have any questions before we start?
Sure. I just know what you told me in the email. Perhaps you could tell me a little bit more about who you are and where you're coming from and why you're interested in all this.
Yeah. Okay. So I'm Ivanka, a high school student from Minnesota, and I'm really passionate about climate change and sustainability. And so I
Mhm.
first got interested in methane emissions when I did my research project at the U of M under a research scientist at the
Which,
USDA.
yeah, be careful, which U of M. So
Oh, yeah, the University of
we're,
Minnesota.
we're going to have to be a little more careful, say Minnesota or Michigan, I suppose.
Yeah, that's true. And then, so I got interested in that. And then I also did a project at Cornell. And
Mhm.
both of these were in reducing methane emissions from livestock using feed additives.
Okay.
And then I started my MethaneMatters initiative to educate people on methane emissions, like their sources, and
Mhm.
then impact and mitigation strategies.
Mhm.
So I started this YouTube channel, and then I also wrote this book that goes much more into detail about methane emissions.
Really?
Yeah, and I'm really excited to hear about your work.
So apparently Mary Lidstrom pointed you in my direction.
Oh, yeah.
So, well, Mary's great. I'm surprised that she didn't agree to do this as well.
Hmm. Yeah.
All right. So anyway, I wanted to ask, so this is what you're doing now. I presume you're a senior in high school?
Yeah.
And what are your future plans?
I think I would like to become a research scientist in
Hmm.
like the climate sustainability space. That's what I'm thinking right now.
But do you have an idea of where you might want to go to school? And when you say climate science, are you thinking more about physical chemical processes or some of the biological interventions that I'll probably talk about and others have as well? I mean, what exactly are you hoping to do? And also, where do you hope to go to study?
Um, I would love to go to Michigan, actually.
Oh,
Yeah,
really?
yeah. And also like Cornell, because I did my research project
Hmm.
there. I'm thinking about UC Berkeley.
Okay.
Yeah,
But can you be a little more specific? Are you interested in the engineering aspects or more in the science aspects?
I think I'm
Oh,
more interested in the science aspects.
mhm.
Um, my research projects have been in like biochemistry, so I think like I'm really interested in that. It's really fun.
Mhm. Okay. No, well, you're preaching to the choir because that's what I do. A lot of my work is basic biochemistry, even though I am in an engineering department.
Hmm,
So I think it's quite fascinating.
that's cool. Okay, so can you share a little bit about your educational background and kind of how you got started?
In the climate space,
Yeah.
well, I got my first degree in civil engineering from the University of Texas at Austin. The reason it's civil engineering is because I wanted to study environmental engineering, but there was no environmental engineering program. It was
Hmm.
subsumed within civil engineering. After getting my bachelor's degree at UT Austin, I then matriculated at Caltech in the master's program in environmental engineering science, and then got my master's and PhD there. Uh, then went to England for a year to do a postdoc at the University of Warwick, where I was just doing more basic molecular biology, biochemistry.
Okay.
And then in '95, started my faculty position here at the University of Michigan in civil and environmental engineering.
Okay. So in environmental engineering, what exactly did you study?
Well, that depends on the level of education. At UT Austin, it was an undergraduate program in environmental engineering. So there's a lot of wastewater engineering, a lot of basic air pollution engineering, just introducing you to the broad panoply of different environmental issues and the engineering strategies that people had designed to try to address them. So water, wastewater, solid waste, air. But then when I got to Caltech, I started working with Mary Lidstrom, who's a very uh, very incredible microbiologist who was studying these weird little microorganisms called methanotropes. And I got really curious about these microorganisms because they can do something that's inherently difficult in that they can consume methane. That's actually hard to do because the carbon-hydrogen bonds are so strong. Just for instance, methane is a wonderful energy source. It's packed with energy. But whenever you, me, or our families, whenever we try to use methane, we can't control the combustion. It goes almost immediately from methane to CO2. But we don't care because this is great. It heats our houses, generates electricity, cooks our food, right? So lots of energy is released. But these microorganisms, methanotropes, if they did that, they blow up. Okay, so they have to control the extent of oxidation. They have to do it very carefully and in numerous steps. And the question is how are they doing that? Because we can't. And so that's where I started working with Mary, was just asking that question. How are these microorganisms able to do this process where they're able to carefully control methane oxidation, where ultimately it does go to CO2, but not immediately? How are they doing all this? And can we understand that and perhaps use it for other purposes?
Ah, that's interesting. So are microbes like your main focus?
Yes.
Okay. So then how did you you got interested in methane, and then what do some of your projects look like now?
Well, you have to understand that I don't typically try to solve a problem in my research.
Okay.
Instead, I try to solve a puzzle.
Okay.
And initially when I got to Caltech, the puzzle is how are these microorganisms eating methane? And as I alluded to before, it's a multi-step process. But the first step is probably the most critical step, where they take methane and insert one oxygen atom from dioxygen and make it into methanol. Okay? That's carried out by a specific enzyme called the methane monooxygenase. And I was just curious, what is this enzyme? How does it do this? Okay? So I started asking basic questions. Okay, where is the enzyme? What does it look like? Can I purify it and can I study it in a simpler system? There must be some way by which the cell makes this enzyme. How does it make this enzyme? Can I find the genes for it? And so I did a lot of work like that. Okay, just basic questions. Okay.
Okay.
And then so I did a lot of work there. And then when we were doing that, we realized, wait a second, this enzyme, its primary substrate is to oxidize methane. But you have to understand, enzymes are basically dumb proteins. They don't have brains. They can't. They'll attack anything that looks like methane. And there are other things that look like methane, like chlorinated solvents, that these microorganisms will attack. And we thought, oh, well, maybe you could use these to help degrade pollutants that we find in various environments. Okay.
Okay.
So that was like, oh, I was more interested in just understanding how the enzyme worked. And then we realized, oh, it could be used for this. So we started studying that, right? But then the systems turned a little bit and people got curious and said, can we use these methanotropes to control methane emissions? I got to thinking about that. And I thought, well, maybe. I mean, certainly they consume methane, but can we design a system where we stimulate their growth and force them to eat more methane? I thought, I don't know. It's a puzzle. So I started looking at that and saying, okay, can we actually create economic, effective systems to harness methanotropic ability to consume methane? And long story short, we did a lot of modeling and we said, yeah, it works, but only under certain situations. Okay, so it's not going to work all the time. You have to be careful where you try to apply it. Okay, so I did that. But one of the more interesting things is that we discovered early on that copper was really important for these microorganisms to eat methane. It's like, okay, why is copper important?
Yeah.
I don't know. At the time, I'm just asking questions, right? Well, again, I'm talking years and years of work here. So it turns out that copper is part of the active site of the methane monooxygenase that enables it to transform methane to methanol. So no copper, dead enzyme. Okay.
That's interesting.
So you got to have copper, right? But then we said, okay, but most cells avoid copper because it's toxic at high concentrations. We actually utilize copper in many situations as a biocide to kill off bacteria. But it turns out these methanotropes, some of them, they'll just keep taking up copper. You give them copper, they'll take it up. They'll just keep eating all the copper. You can just keep on adding more and more and more copper, and they'll just keep taking it up. And they'll turn black from so much copper inside themselves. It's like, That's weird because they survive in copper concentrations that would kill practically everything else. It's like, okay, what's going on here?
Yeah.
So how are they collecting copper? You know, how are they controlling the toxicity of copper? Because copper is inherently toxic for reasons I don't want to go into here. So did a lot of work there and said, oh wow, they make this compound that binds copper. Okay, and it's called methanobactin, and it binds copper really strongly. And here's where it gets really interesting. I was working with a guy named Alan DeSpirito at Iowa State on this, and published a lot of work on this compound. The structure, the genetics, and then somebody from Germany contacted us. His name's Hans Schischke. He said, I study a disease called Wilson's disease. And I said, okay, what's Wilson's disease? Because I didn't know. It turns out that all of us need a little bit of copper in our diet, okay? Because there are certain enzymes that we have that require copper for their activity. Okay. Unfortunately, some people lack the ability to remove excess copper, okay? So if you eat certain foods that are high in copper, so if you like lobster or chocolate, those are naturally high in copper. And a normal body would recognize, oh, there's too much copper being taken up. So what happens is that a normal body would collect the excess copper in the liver, okay? And then that excess copper is pushed into the bile and then it's defecated out. So it's a way of just reducing the overall copper levels in the body, okay? To keep them at the level that it needs to be. Yeah. So with people with Wilson's disease, they collect the copper in the liver, but they can't push it out. So it just builds up, gets higher and higher and higher over time. And if it's not caught soon enough, it can lead to irreversible liver failure. But you can die from this disease. Okay.
Okay.
And it turns out that there's no cure.
Oh.
There's just therapies that prevent. Once someone is diagnosed with this disease, there are certain therapies that are approved by the Food and Drug Administration that just prevent further damage. Doesn't repair damage that has already occurred. Okay. And so people have to be very careful once they're diagnosed. They have to have a copper-free diet. They have to take this medication several times a day. It typically causes gastrointestinal distress, and so people don't like taking it because it makes them feel uncomfortable. But anyway, to get back to this compound that the methanotropes make, Hans Schischke asked for it, and Alan DeSpirito gave me some, and I thought that would be the end of it. But then Hans contacted us sometime later and says, it works. It actually removes copper from the liver. He tested this out in rats. And so he has rats have this disease, and he injected them with this compound, methanobactin, and it actually removed the copper from the liver and put it in the bile, and the excess copper was defecated out. Somehow it replaces the broken pump that the liver should have. Okay? And so now we're doing a lot of medical development, drug development, trying to see if we can't scale the production of this compound up and enter into clinical trials to see if this helps people.
Okay, that's really cool.
But so it's just starting asking simple questions and then just following your nose to see where it takes you. Okay.
So have you done are there any other industries you're working in, like agriculture or anything?
Yes. We know that copper is important for methane removal. It's really important for methanotropes. And they make this really interesting compound to collect it. Okay? And then again, it's called methanobactin. Working with one of my former students, his name is Sakwon Yoon, he realized that there are other environmentally very important enzymes that require copper. And one of them is called the nitrous oxide reductase. Okay. Now, I don't want to go too far into the weeds, but methane's a potent greenhouse gas, about 30 times more potent than CO2. But nitrous oxide is also extremely important. Okay. It's about 300 times more potent than CO2. Now, there's different microbial activities where nitrous oxide is an intermediate of respiration. That is, there's what we call nitrate respiring microorganisms. These if there's no oxygen, these guys don't care. They'll dump electrons to nitrate, and what they do is they take in nitrate, reduce it to nitrite, nitric oxide, to nitrous oxide, and then ultimately to dinitrogen. Okay, so it's a multi-step process. That last step where you convert nitrous oxide to dinitrogen is carried out by the nitrous oxide reductase that requires copper. Okay, so we were just asking the question, what would happen if you expose denitrifiers that would normally take nitrate all the way to dinitrogen? What if you expose them to methanobactam? What would happen? Turns out that methanobactam binds all the copper, limiting the ability of the denitrifiers to collect it, and so denitrification stops at nitrous oxide.
Oh, that's interesting.
So it is reduced. You want it to go reduce all the way to dinitrogen. It stops at nitrous oxide. So we've done this in many different systems, in pure cultures and in soil samples. But the take-home message here is that if you stimulate methanotrophs to try to enhance methane removal, you could reduce. You could actually reduce the nitrous oxide reductase activity, and so you could actually increase nitrous oxide levels. So you could actually make the problem worse. By stimulating methanotrophs, they produce more of this compound, they collect more copper, and starve the other guys for copper. And so yeah, you remove methane, but you blow out nitrous oxide.
So do you have any ideas on how to solve that?
Sure. It's actually, if you think, it's one of these things. You just have to make sure that everybody's happy. Right?
Yeah.
So you just have to make sure that there's enough copper to go around.
Okay.
So what you can do is, I'm working with people at UC Berkeley on this issue, where we're just saying, okay, what if we add copper? Can we get the best of both worlds? Can we enhance methane oxidation while also limiting nitrous oxide production? And the fast answer is yes. If you make sure that everybody has enough to get by, then everybody's happy, right?
Yeah.
So we're proposing that going forward, currently right now, when we have agricultural processes, they just slap, particularly for corn and particularly here in Michigan, they just slather on the ammonia, right? And the nitrate. You slather on that nitrogen to really to get a kickstart in the plant growth. But when you do that, you will induce a lot of denitrification. And so you could inadvertently have substantial nitrous oxide emissions. But here's what we say. Instead of just adding the nitrogen, add a little copper as well to make sure that everybody's happy.
Okay. So you mostly focus on like reducing methane emissions from the source, like stopping it from being produced, or do you do some like methane trapping or removal also?
Well, no, I don't try to stop it at the source, because that would be trying to limit methanogenesis. Because as you know, that there are other microorganisms that produce methane. So I don't look at that side of the problem or the coin.
Okay, yeah. So it is removal. Okay.
It is removal, yes. Okay.
That makes sense. Yeah. Are you doing any other cool stuff?
Well, let's see. Well, I think it's cool because it's what I do. I don't know if your audience might find it interesting, but I'm now doing a lot of metabolic modeling of methanotrophic activity, because what we're just trying to figure out is, methanotrophs pull in methane, they pull in nitrogen, they pull in phosphorus, they pull in sulfur, they pull in all these trace nutrients that they need, and there's all sorts of different reactions that occur inside the cell, and these things are combined and manipulated to make all the different subunits of the cell, the DNA, the RNA, the different proteins, blah 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 blah. And what we're trying to do is come up with basically a big complex model that describes how things are taken in and how the carbon and these other nutrients are transformed to make whatever the cell needs. It's a big mathematical problem. And the idea is, okay, fine, if we can understand how the cell is using all these things and where it's forcing carbon to go or nitrogen to go or whatnot, can we then manipulate the metabolism so that we can force other things to be made that might be more beneficial for us, right? Because the cell is eating methane to make more of itself. But we may say, hey, we want you to eat methane to make, say, more of this putative drug, right? Okay. To make, say, more methanobactam, or to make more lipids, because we can use lipids as a precursor for biodiesel. Okay. So I'm doing that. But again, that's mostly it's a lot of mathematical modeling, but then we run the model. We say, okay, fine. What if we knock out this gene, or we play with the system where we add in these reactions or these enzymes? What does the model tell us? What is it going to say? Are we going to make anything useful or not? Right? And then we take, if the model says, yeah, if you do this, you should be making a lot more of that. Then we say, okay, let's do it in the lab and see if the model's right.
Yeah, that's really interesting. So then you don't have to test it like in real life in the lab.
Yeah, because genetically manipulating microorganisms, particularly these microorganisms, can be time consuming.
Yeah.
Okay? This is not E. coli. You can do genetic manipulations of E. coli in a day. Here it takes a couple of weeks. And so it saves a lot of time. So we can say, okay, fine. Of all the different opportunities or different options that we have, which one do we think is going to be the most successful? And let's try that first.
Okay. Yeah, I actually talked with another scientist, Dr. Ruckel from the University of Iowa, and he does simulations and modeling like this to like test which compounds could reduce methane emissions.
Yep. So that's another strategy. Yep. So there's a lot of people do metabolic modeling like that. It's getting to be quite popular.
Do you use any other technologies in your research?
Well, a lot of different genetic tricks to remove or add in different genes to control gene expression. So a lot of molecular biology tools. I use a whole suite of analytical tools. Different spectrometric techniques to characterize metals or metabolic intermediates. So, I do things like high performance or high pressure liquid chromatography, gas chromatography, mass spec analyses, the whole suite. These are fairly common tools that pretty much anybody in the microbiology lab would use.
Okay. So what does your team composition looks like? Who all is in your lab?
Well, right now it's a little small. Earlier this year, I had a lot of undergraduates and some graduate students and a postdoc. The graduate students have graduated and gone off, hopefully to do great and wonderful things on their own. And so I have a postdoc and a couple of undergraduate students right now. I'd like to get more graduate students, but as you're probably aware, the current funding climate is a bit tentative, to be polite.
Yeah.
And here at the University of Michigan, just something for you to know. If we admit a PhD student into our labs as faculty, we are obligated to commit 4 years of support to them.
Oh.
Right?
Okay.
So let's make sure that they have adequate support to finish their degree. And right now, I don't have 4 years of funding sitting around that I can use to support an incoming PhD student. I do have some grants in review, and if one or more of them is selected for funding, then I'm going to start recruiting more students. But given the uncertainty as to the availability of funding right now, I've decided to wait a little bit before I try to replace my graduate students.
Yeah, that makes sense. So do these people come from biology backgrounds, or what backgrounds do they come from?
All around. I've had engineering students. I've had students with civil engineering degrees in my laboratory, with environmental engineering degrees, because I got my PhD, I got my bachelor's degree 37 years ago, and then environmental engineering wasn't really its own discipline. It wasn't its own. Most colleges did not have a separate degree program in environmental engineering. It's just civil engineering. But now a lot of programs have environmental engineering degrees. So I have civil engineering students and more specifically environmental engineering students, chemical engineering students. I've had a lot of chemists and biologists. And I've even, perhaps the oddest student that I ever had was a student that had a bachelor's in public accounting.
In what?
Public accounting.
Oh, interesting.
Yeah, he was a CPA, certified public accountant, and worked as a CPA for quite some time. Decided he wanted to go back to school and do something different. And so he worked in my lab for a couple of years. So I get students from all disciplines. So.
That's cool. So what are the qualifications that someone would need to have if they wanted to join your lab?
Well, the first and foremost is they have to be curious. And by that I mean you have to read. Just ask questions, read the literature, find out what other people are doing. Ask questions all the time. Say, oh, when I started off, I'd read papers and wouldn't understand half of it. And I would just ask other people, more senior graduate students or postdocs or professors. And I say, I got to this point. Then I got lost. What did they do? And what does it mean? And so just asking questions, read. And then as you read more, you start to understand more. And then you say, oh, okay, this paper, I'm not so sure if it actually proved what it said it proved. Because I don't think that the techniques they used were really the right ones. I think that there might be some artifacts in their data that bias their results, and so that they're not making the right conclusions, right? So you get better at it, right? So you have to be curious. Just ask questions and follow your nose, right? Along with curiosity, it's just the problems that we're trying to address now are just so big. You can't do it all alone. You can't know everything. It's impossible. So you're going to have to collaborate with people. Yeah. So you're going to have to develop a network where you work with not just other, say, in my case, microbiologists, but you have to work with soil scientists and environmental engineers, reactor engineers, and sometimes you have to work with policy people to make sure that the policy people can help communicate to the decision makers why this should be done. Because that's not my expertise. I can say this should be done, but how do I communicate that effectively? So you have to collaborate with people.
Yeah.
And you also have to communicate. You have to be clear as to what you're doing so that everyone understands. So you have to have really good communication skills, both orally and in writing. And you have to be willing to put yourself out there. You have to be confident and say, I can help. Okay. But then show that you can help, right? So that's what I would recommend. Be curious, be collaborative, and make sure that you can communicate.
Okay. And then you mentioned you collaborate with someone from UC Berkeley. So what other universities have you collaborated with, or do you?
Well, my single biggest collaboration is with Iowa State University.
Okay.
Because that's one of my closest colleagues, a man named Alan Desperto. He is a faculty member there. He and I have published, I've lost count, but I think we've jointly published something like 50 papers.
Wow.
University of Warwick, University of East Anglia. I've also worked with some Russian universities. I've collaborated with the University of Vienna.
Okay.
I've collaborated with Korean universities, all around. It's just either I reach out and say, hey, I'd like to work on this project. You seem to have some skills that could be helpful, or vice versa. They reach out to me, saying, hey, I'm working on this project. I think you could help. And, usually I say, sure. If I think I can contribute, I'll say, sure. Right? Sometimes I say no if I don't think that I can. If they're looking for expertise that I don't have, and I just say, I don't think I can help you too much, but I'll try to point them in the right direction. Okay? So all around. Yeah, just whoever.
Yeah, I'm curious. Have you collaborated with anyone in India?
Yes and no. One of my favorite postdocs was from India. She went back to India, and she's working at I am so bad. She's working at one of the Indian Institutes of Technology, but I can't remember which one. Okay. So yeah, we keep in touch and share ideas back and forth. So that's the most extensive interaction that I have with Indian universities at the moment. So
that's cool. Okay, so this is kind of related to the last question, but do you have any advice for a high school student or just someone like me who wants to go into this space as a career?
Well, like I said, be curious, read. Just don't do the minimum. Seek out opportunities to learn. It's more and more common now than it was, say, 30, 40 years ago to have undergraduates in laboratories. It's a wonderful resource that people are starting to recognize. So I would say if you have the chance, approach a faculty member who you think you can work with and ask questions. What are you doing? And if it interests you, say, are there opportunities? They may give you some simple tasks to begin with, just to make sure that they can trust you, that you're not going to be a danger to yourself or someone else. Because the nature of my work is that we work with hazardous chemicals all the time. And so you just have you have to make sure that people understand that there are specific procedures that you have to follow, and that they have to have you have to follow certain documentation practices, because you have to say what you did, how you did it, and how you disposed of it. So once we start to develop more confidence in them, we give them more and more responsibilities. So I would just say, be curious, ask questions, do more than the minimum reading in class, ask questions in class. I would say when you're in if you don't understand something, stick out your hand and ask.
Yeah.
Don't be ashamed. Don't be bashful, because I guarantee you, if you don't understand it, there's probably a fair number of other students that didn't understand it.
Yeah.
Okay, and so you're helping everybody out by asking the question, and that actually raises your profile to the faculty member. Say, oh, this student is engaged. They want to learn. They're asking good questions, and so that kind of feeds on itself. And they may be the one to initiate and say, would you like to do some projects with me?
Yeah.
Okay, so I would say easy peasy. First thing, ask questions in class. Okay, don't be afraid.
Okay.
Do more than the bare minimum. If they say read these pages, in the textbook, okay, you can read that and say, okay, well, I'm not sure if I really understand it, because maybe the textbook didn't describe the concept very well. Then you can just do a Google search and say, okay, tell me more about this particular topic. And then you can hopefully you can build up your understanding. Okay.
Yeah.